basically the first step of protein synthesis is transcription, right? And transcription occurs inside the nucleus because the DNA cannot exit the nucleus, and DNA basically has the code, has the instruction to produce or to formulate a particular protein. So for that, we need a messenger, we need a mediator, which can take that code to the ribosomes, and then ribosomes would translate that code and form a polypeptide. So we understood that transcription occurs within the nucleus and translation then occurs upon the ribosomes. ठीक है जी? Then we also understood that some ribosomes are present on the rough endoplasmic reticulum, giving it a rough appearance, a granular appearance, due to which it is called rough endoplasmic reticulum. And this we also understood is continuous with the nuclear membrane. Why? Because it's just easy to have something which has the ribosomes on it, where the translation process has to occur, where the mRNA from the nucleus would leave and then attach to the ribosomes. So if that site of translation is closer to the nucleus, it's just that the mRNA would find it easier to attach itself to the rough endoplasmic reticulum and translation would occur. We also understood that because rough endoplasmic reticulum has ribosomes attached on its surface, translation can also occur on the rough endoplasmic reticulum, and therefore our rough endoplasmic reticulum is also the site of translation. So it is involved in translation. Why? Because it has ribosomes present. So till yesterday, we covered all of this. I assume that everything is clear so far. So now, what else can rough endoplasmic reticulum is responsible for? So one of the main salient features of rough endoplasmic reticulum is protein folding. Now, what do we mean by protein folding? Let's just understand that in a bit. Okay. So for example, we understood that ribosomes made a polypeptide chain. So basically, we have a ribosome over here. ठीक है जी? We have a ribosome, and this ribosome coded for a polypeptide. Now we have this polypeptide, right? Now, what is the process that we're studying? We're studying the process of photosynthesis, right? So in photosynthesis, sorry, I'm saying photosynthesis, I meant protein synthesis. We're studying protein synthesis, right? हमें क्या बनाना है? हमें protein बनाना है. And we know that protein is basically made up of many polypeptide chains. When they interact together, when they formulate together, a protein is formed, right? So now, these polypeptides have to combine together to form what? To form a protein. Till now, we have only formed polypeptide. कैसे हमने polypeptide बनाया? We had a DNA. It had a particular gene which was coding for a particular polypeptide. How? We had an mRNA that was leaving the nucleus, attaching itself to the ribosome on the RER or in the cytoplasm and forming a polypeptide. Now we have hundreds of genes and we have number of mRNA being formed simultaneously within the cell. ठीक है जी? And these polypeptides, we have multiple polypeptide chains, right? So for example, 20 polypeptides combined together. This is an example that I'm giving you. Combined together to form a protein. Now, this entire process, that 20 polypeptides are coming together to form a protein. So definitely हो सकता है हमारे पास बहुत सारी जो पॉलीपेप्टाइड की चेन है वो ऐसे बहुत सारी इस तरह से we have a lot of polypeptide chains. Obviously, a certain orientation, a certain dimension, a certain involvement, a certain attraction, interaction is necessary. So I cannot just join all of these together like this. इस तरह से तो वो join हो नहीं सकती. There has to be a proper mechanism, a proper science behind all these polypeptide chains to come together and form one unit of a protein, right? So this entire process, when a lot of polypeptide chains which are formed as a result of translation, when they combine together to form a protein, This entire process is known as protein folding, which is a lot of polypeptide chains combined together. They fold onto one another in a set orientation to form a protein. And obviously, this process, the process of protein folding, can only occur once we have multiple polypeptide chains. And where are these polypeptide chains coming from? These polypeptide chains are coming from as a result of translation. Because translation has occurred on rough endoplasmic reticulum, and even if it has occurred in a freely suspended ribosome, all these polypeptide chains would come onto the RER. This is your RER, rough endoplasmic reticulum. And now this rough endoplasmic reticulum will fold all these polypeptide chains. I'll just open my video. Okay, so that you can clearly see. Okay, so all these polypeptide chains would now come together in a certain way that these polypeptide chains will fold onto one another and they'll form a particular protein. So this entire process occurs after translation. ठीक है जी? So we call this, another name for protein folding is also post-translation modification. So this is called as post-translational modification. Why? Because we need polypeptide chains to fold together, and once we have these polypeptide chains once translation has completed, we have the polypeptide chains. Now these polypeptide chains, once the translation process is complete, they are modified, they are converted, they are folded to form a particular protein unit. Okay. So RER is also responsible for post-translation modification or protein folding. Now once a protein is formed, हमारे पास सारी polypeptide chains को हमने modify कर दिया, हमने properly fold कर दिया, we have a protein at our hands. Now this protein has to undergo some more modification. It has to undergo some more processes to be properly functional. So now what RER does, once it has formed this protein, once the protein folding is complete, once post-translational modification is completed, what happens is that we would now, the RER would basically package this protein. It would package this protein and it would also transport that protein to another cell organelle, which is the Golgi body. So basically what is happening in the RER? RER is involved in translation. Why? Because it has ATS ribosomes on its surface. Those ATS ribosomes are basically converting or translating the messenger RNA into a polypeptide chain. Multiple polypeptide chains come together to form a protein. They're folded, they're organized, they're modified in a way that a protein is formed. This process is the process of protein folding, post-translational modification. And once we have our protein ready, what we would do is the RER would package these proteins into small vesicles, and these vesicles would then be transported to another cell organelle present within the cell near to the rough endoplasmic reticulum, which is called the Golgi apparatus or the Golgi body. Any questions so far? Is it clear? Okay, what do the vesicles do? Now, okay, so for example, let's have a look over here. This is our cell. ठीक है जी. This is our cell. We have our nucleus over here. ये उसका nucleolus है, right? Now, around that nucleus, I have my rough endoplasmic reticulum, okay? I'm drawing the ribosomes on it. Okay, so this is my rough endoplasmic reticulum. Now, what is happening over here? Let's have a look at this ribosome. For example, this ribosome just made one polypeptide chain. Okay, this is the polypeptide chain which this ribosome has just made. Okay, now this polypeptide chain is made. This ribosome has made this polypeptide chain. this ribosome has made this polypeptide chain, and all these ribosomes obviously make simultaneously a lot of polypeptide chains, a multiple protein synthesis reaction are occurring at a given time, right? एक वक्त में हो सकता है thousands of polypeptide chains are being formed on one single RER, or thousands of ribosomes are formed within a cell. Sorry, thousands of polypeptide chains are formed within the cell, right? So now, once the process of translation is complete, what happens is all these polypeptide chains then undergo a process of protein folding within the rough endoplasmic reticulum. So, for example, all these proteins, all these polypeptide chains are folded, are organized, are modified to form a protein unit, and it looks like something this. Okay, within the RER did the post-translational modification, folded all those polypeptide chains, formed this protein unit, and now what it does is, now this protein unit is inside the rough endoplasmic reticulum. We are still left for some processes to be completed. Okay? Now, this protein needs to exit the rough endoplasmic reticulum. Now, it cannot just throw away this protein, right? It cannot just eject out or secrete this protein just like that. So, what RER would do, for example, just take this analogy that you have to give a present to someone. Now, for example, you got a gift for someone, and you have two ways. You take that gift, you do not wrap it, you just hand it to that person just like that. Or the other thing that you would do is you would wrap that gift into a nice packaging, and then you would hand it over to the person who you are gifting that gift, right? So what rough endoplasmic reticulum does is that once our protein is formed, it would package that protein into a vesicle. It would properly package that protein into a vesicle. एक proper boundary में उस protein को it would package, and then what it would do is, okay, so basically what happens is, I'll just properly draw it out. So now it has packaged it into a vesicle, okay, and now this vesicle would be transported to something else. So it cannot directly leave or eject that protein. It has to neatly package it. It has to surround it by something, and then it sends it over to the Golgi apparatus. Now the Golgi apparatus would be lying somewhere near to this RER. So this is our Golgi. Let me draw it out. So we have our Golgi apparatus somewhere near the rough endoplasmic reticulum. So it would basically now package and transport that vesicle containing the protein inside it to Golgi apparatus. And here Golgi would modify this protein more. So now we'll understand how this protein would undergo more modification when it reaches the Golgi apparatus. Is it clear so far? Does that answer your question? Okay, perfect. Now, let's understand what is the role of a Golgi body. So let's first understand the structure of a Golgi body. So Golgi body is a stack. Now what is a stack? हमारे पास, when we were studying rough endoplasmic reticulum yesterday, we said that a rough endoplasmic reticulum is an interconnected system of flattened membranous stack. Now we outlined over here that how all these membranes are connected to one another. They are not discontinuous, they are not separated from one another. All these membranes, all these layers, which are called cisternae, these membranous sacs are interconnected. However, if you look at the Golgi that I drew in this cell diagram and the Golgi that I have pasted over here, you would notice that yes, there is a system of membranes, but they're not interconnected. Every single layer, every single membrane is separate from one another. There is no connection. They are stacked upon one another. Just for example, how I would I have this one book and then I have this other book. So I have stacked these books to one another and they're not connected. I've just piled them up upon each other. So the same way in Golgi, unlike an endoplasmic reticulum, the membranes are not continuous. They're not interconnected. They are stacked upon each other. They are flattened membranous sacs, and these membranous sacs are also called cisternae. But the difference between a Golgi body and an endoplasmic reticulum is that these membranous sacs are not continuous, are not connected to one another. They are stacked upon one another. Okay. And now, if you could notice that each membranous sac has a swelling on its end. So if you can see, it's swelled from this end. It is swelled from this end. तो इसका structure Golgi का कुछ इस तरह का होता है. So any question, and also Golgi does not have ribosomes on its surface. Ribosomes are only present or attached on the surface of only one cell organelle, which is the rough endoplasmic reticulum. So the differences between RER and Golgi apparatus are two. One of them structurally, process के accordingly, because दोनों के functions very different from one another. But when we're talking about the structure and we're observing their structure under a microscope, we would observe that the two major differences between the structure or the appearance of a rough endoplasmic reticulum and the Golgi body are two, which are that the membranous sacs, the cisternae, they in RER, they are continuous, they are connected to one another, they are not separate. But in a Golgi apparatus, those flattened membranous sacs are disconnected to one another. They are stacked upon one another. They are piled up upon one another. And another difference is that RER has ribosomes attached to its surface, and the Golgi apparatus does not have ribosomes attached to its surface. Okay, Ji. Now coming to the functions of a Golgi body. Now, Golgi body is responsible for protein modification. It is responsible for what? It is responsible for protein modification. Now, you do not have to confuse this with post-translational modification, because post-translational modification में क्या हो रहा था? In post-translational modification, we were basically making, or we were folding polypeptide chains into a protein, because protein is a macromolecule, and we know that a macromolecule or a larger molecule is made up of smaller units, smaller sections. So our proteins are made up of multiple polypeptide chains. So in post-translational modification, we were just folding those polypeptide chains into a protein. Now, once the protein making part is limited to the rough endoplasmic reticulum. In the rough endoplasmic reticulum, the polypeptide chains are made, and then the polypeptide chains are then converted into protein. Now, the protein is fully made within the RER. So the protein synthesis process is complete in the RER. We have made polypeptide, and then we have also converted, we have modified those polypeptide chains into a proper protein unit, into a proper macromolecule. So our protein synthesis is complete at the end of the within the RER. Now in Golgi body, we are modifying that protein. The protein we have made in the rough endoplasmic reticulum, हमने जो protein already बना ली है, हम उसको modify करेंगे कहाँ पर? Golgi body में. We are not doing protein folding over here. Complete protein हमारे पास बनी बनाई आ गई है कहाँ पर? In our RER. RER has done the protein synthesis, the protein making part for you. Now, once the protein is received to the Golgi body, it is saying, okay, let's just modify it more. For example, you can say you had a gift, right? What RER did was it had a present for Golgi body. It wrapped that present with a gift wrapping sheet. Now it has given that to a Golgi body. Now Golgi body is like, okay, I've got the present, I've got the protein, it's packed and everything. Now let's make it more beautiful. Let's add a ribbon to it, let's add a card to it. So you have the gift. RER has given you the complete gift. It has given you that protein. But now once Golgi receives that protein, it goes on like, let's modify it more, let's add more things to it, let's make it more interesting. So that is the rule of a Golgi body. Now what happens is protein modification is कि हमारे पास बहुत तरह के molecules होते हैं. हमारे पास conjugated molecules होता है. Now, what do I mean by conjugated molecule? One molecule, we have a molecule and then we add another different molecule to it. For example, I have a protein. Now, where is this protein coming from? The Golgi is receiving this protein from RER. ठीक है जी. RER sent this protein to Golgi. Now, what Golgi did? In Golgi, the protein that it received from RER, it added a chain of fatty acids to it. It added lipids to it. Now, protein, उसने कहाँ से receive किया था? RER. A complete protein it received from the rough endoplasmic reticulum, and what it did was it added a lipid chain to it. Now, it is a protein as well as a lipid. So now this protein is not the same as a normal protein. You would say a normal protein is only and only comprising of protein units. A normal protein or a simple protein would never have lipids upon it, right? Now, this is something different. This protein has been modified to something totally else because it now has a lipid chain attached to it. So now what it would be called? It would be called a lipoprotein. Why? Because it has a lipid as well as protein. Subsequently, when we would study something like a peptidoglycan cell wall. The peptidoglycan cell wall is not an example of protein modification, but basically you are adding a peptide chain to a carbohydrate over there. So that's also a conjugate molecule that you're combining protein and carbohydrates. But let's just come back to protein modification as of yet. In protein modification, does it always? No, it can add anything else. Let's just come to another example. For example, there's a protein and it adds a carbohydrate molecule to it. Let me just shift it a little bit. We have protein and the Golgi just added carbohydrate to it. So now this particular protein is no longer a normal protein for you. It is a protein with a carbohydrate unit attached to it, and it would be called a glycoprotein. Acha. Now the thing is, the catch, it's not something that you should know, but just for your information, basically how, for example, there's a family and it has multiple kids, right? And every kid would have a different name, but their surname would be the same, or their father's name would be coming at their end. So the molecule which is coming at the end, for example, protein, दोनों में protein is the surname, protein is coming at the end, glycoprotein and lipoprotein. And if you could notice, this is a प्रेफिक्स. हमने कोई प्रॉपर कार्बोहाइड्रेट का पूरा नाम नहीं लिखा, और लिपो में भी हमने पूरा लिपिड नहीं लिखा, हमने खाली लिपो लिखा है. तो इसका मतलब क्या हुआ? दैट आवर मेन मॉलिक्यूल इज दिस मॉलिक्यूल, जिसका पूरा कंप्लीट नेम जो कि एंड में आ रहा है वो हमारा मेन मॉलिक्यूल है, और इस मॉलिक्यूल पे कोई दूसरा मॉलिक्यूल आकर अटैच हुआ है. So the molecule which is acting as a surname is the main molecule upon which something else which is in the prefix is coming and adding or attaching itself onto it. Okay? And that's the reason why I said that peptidoglycan is not an example of protein folding. Why? Because peptide, protein part, peptide हमारे पास proteins होता है ना? Peptide is a prefix over there. It's not a surname over there, right? So it means that glycan. Now, anytime you see glyc, it's basically carbohydrates. So basically, carbohydrate is our main molecule over there, and a peptide chain is coming and attaching to it to form the peptidoglycan molecule. So basically, in protein modification, which the Golgi does, what it does is that it attaches or it converts the protein into something different. Okay? So protein is modified. That protein which it was receiving from the rough endoplasmic reticulum is modified into something else. Okay? So this is basically protein modification. Now, once this protein is now new, अब इस protein में जो भी उसको add, subtract, changes उसको करने थे, once it has formed this molecule, once it has completed the protein's modification, it would now the entire process, हमारे पास protein synthesis भी हो गया, protein modification भी हो गई. We have not only formed a protein in the rough endoplasmic reticulum, now that protein is completely modified, and this final product, now the final product is basically formed in the Golgi. हमारे पास once the protein modification is done, we have our final product, and then that final product obviously, अब वो चीज हमारे पास वो जो molecule है, ये enzyme हो सकती है, ये कोई hormone हो सकता है, ये हम हार्मोन यहाँ पे प्रोटीन, हमारे पास जो पेप्टाइड हार्मोन्स होते हैं ऑब्वियसली खाली वो ही हमारे पास यहाँ पर आ सकते हैं. बेसिकली वी हैव टू टाइप्स ऑफ हार्मोन्स. वी हैव स्टेरॉइडल हार्मोन्स. स्टेरॉइडल हार्मोन्स आर लिपिड बेस्ड हार्मोन्स, एंड वी हैव पेप्टाइड हार्मोन्स व्हिच आर ऑब्वियसली प्रोटीन बेस्ड हार्मोन्स. तो हमारे पास जो प्रोटीन बेस्ड हार्मोन्स होते हैं वो ऑब्वियसली इसी प्रोसेस के थ्रू बनते हैं कि हमारे पास पहले प्रोटीन सिंथेसिस होता है. वंस दैट प्रोटीन सिंथेसिस इज कंप्लीट, that protein is then sent to the Golgi vesicle. That Golgi vesicle completes its modification, whatever has to be added to it, whatever has to be subtracted from it, all of that is done within the Golgi, and then we have our final product. And now this final product, या तो वो हमें intracellular use करना होगा कि सेल को उस प्रोटीन की जरूरत होगी, या फिर उस सेल को वो जो प्रोटीन बना है गोल्जी के अंदर, जो पूरा प्रोटीन सिंथेसिस का प्रोसेस हुआ है, उस फाइनल प्रोडक्ट को सेल के बाहर भेजना होगा. So in any case, this final protein, after the modification process has been completed, it needs to get out of the Golgi. Why? Either the cell needs to use it, or it has to be exported out of the cell. So in any case, this modified protein needs to get out of the Golgi. So all these proteins which are modified, they are now packaged by the Golgi body. ठीक है जी? These proteins, once their modification is complete, the Golgi packages them. And how does it package? Again, Golgi makes vesicles of it. And these vesicles, जैसे कि यहाँ पर आप देखो, ये एक vesicle है, ये एक vesicle है. All these vesicles are pinched out. जो ये swelling है ना हर उसके end पर, basically होता क्या है? For example, imagine हमारे पास, आप एक स्नेक की स्किन को इमेजिन कर लो, या अभी सर्दियों में हमारी भी जो स्किन होती है वो बहुत फ्लेकी हो रही होती है, वो बहुत ड्राई हो रही होती है, तो वो पिंच ऑफ हो रही होती है. उसी तरह से जो ये एंडिंग्स है ना गोल्जी की, इन एंडिंग्स की जो स्वेलिंग है, बेसिकली यहाँ पर हमारे पास वेसिकल बनता है, और फिर जब वो वेसिकल कंप्लीट हो जाता है, वो स्वेल होते रहते हैं, होते रहते हैं, होते रहते हैं, फिर वो जब स्वेल होके कंप्लीट अपना एक सर्कल फॉर्म कर लेता है ना इस तरह से, वो स्वेल हुआ 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 हुआ, once it is now a complete circle, it would pinch off like this. तो वो pinch off खुद ब खुद एंड से हो जाएगा, और फिर एक नया प्रोटीन आ जाएगा, और फिर वही जो पूरी साइकिल है वो रिपीट होगी. So that is why each membranous sac has a swelling on its end. Why? Because the vesicle, once the protein modification is complete, वो एक vesicle बनाएगा अपनी, और उसमें वो जो vesicle होगी वो pinch off कर जाएगी एक end से, और फिर वो जहां पर अगर इंट्रासेल्युलरली रिक्वायर्ड होगी, तो जिस भी ऑर्गेनील को उस प्रोटीन की जरूरत होगी, वो वेसिकल उस ऑर्गेनील के पास चला जाएगा. और अगर ये प्रोटीन, ये वेसिकल सेल के बाहर जानी है, तो वो सेल मेम्ब्रेन के पास जाएगा, और सेल मेम्ब्रेन से फिर वो एग्जिट कर जाएगा सेल के बाहर. एनी क्वेश्चन सो फार? कोई क्वेरी, कोई चीज जो समझ नहीं आई हो? Okay, perfect. Now the third part. Golgi is also responsible for the production of lysosomes. Now, when we were studying the ultra structure of an animal cell, और हम उस सब organelles को label कर रहे थे, so I said that lysosome are a specialized type of Golgi vesicle. And what do I mean by that? Now, Golgi vesicles, हर तरह का हमारे पास जो प्रोटीन बन रहा होता है, रफ एंडोप्लाज्मिक रेटिकुलम सारे प्रोटीन्स बना रहा होता है. All vital proteins, be it our enzymes, be it our peptide hormones, be it any type of protein which is required by our body, all these proteins are formed in what manner? कि nucleus mRNA बनाता है, mRNA goes to a ribosome, ribosomes forms the polypeptide chains, the polypeptide chains are then folded, undergo post-translational modification within the RER. We have our protein completely formed in the RER. That RER sends that protein to the Golgi body. That Golgi body modifies that protein, and then it forms a vesicle, and then the vesicle is out of the Golgi body. Now, बहुत सारी multiple चीजें, multiple तरह के proteins, multiple तरह के enzymes, multiple तरह के hormones बन रहे होते हैं Golgi के थ्रू. एक पर्टिकुलर वेसिकल होती है लाइसोसोम, और उसकी एक अपनी फंक्शन होता है, उसकी एक अपना रोल होता है, तो वो भी गोल्जी बना रहा होता है. ऑब्वियसली लाइसोसोम अगर एक वेसिकल है तो उसके अंदर प्रोटीन ही होगा. व्हाई? because Golgi is forming what vesicles? Golgi is forming vesicles which are containing modified proteins, right? So lysosomes would also have protein molecules inside it. So lysosome is one of the hundreds of vesicles which Golgi is making. तो वो जो स्पेशलाइज टाइप की ये लाइसोसोम वेसिकल है जिसको हमें बाय नेम बाय फंक्शन पढ़ना है, उसका नाम होता है लाइसोसोम। एंड ये भी कौन सिंथेसाइज कर रहा होता है, ये भी गोल्जी फॉर्मुलेट कर रहा होता है। ओके, any questions so far, or should I just move on to our next topic of lysosomes?